Welcome friends. Today we will discuss about prokaryotic cell structure. I have highlighted the important points for you to remember. So let's begin. So these prokaryotic organisms, they are the most abundant organisms on earth. Okay, and they are divided into two distinct groups. What are the two distinct groups? One is bacteria, which is also known as eubacteria, and the other is archaea, also known as archaeobacteria. Now also remember, these prokaryotic cells, they do not have a membrane bound nucleus. Okay, nucleus is there, but true nucleus is absent. Now we are going to study all these points in detail. We are just doing the overview right now. Cell structure. So all prokaryotic organisms, they are surrounded by plasma membrane. Okay, they do not have membrane bound nucleus as well as they do not have any subcellular organelles, but they have infoldings on the plasma membrane. Okay, these plasma membrane have infoldings. Okay, these infoldings are known as mesosome. Okay, and this DNA which is present in the prokaryote, they are condensed within the cytosol to form nucleoid. Okay, if this is a cytosol, the DNA are condensed to form what? To form nucleoid. Now coming to the cell wall of prokaryotes, they are made up of peptidoglycan. The composition of peptidoglycan is protein and oligosaccharide. Okay, and the cell wall helps in protecting the cells, prokaryotic cells, from the mechanical and osmotic pressure and the antibiotic penicillin. We already have heard about penicillin. These penicillins, they target the enzyme. So penicillin is an antibiotic. What it does, it targets the enzyme which is responsible for cell wall synthesis. Which enzyme does it target? The enzyme which is responsible for the synthesis of the cell wall. Okay, now there is a little difference in the cell wall of gram-positive and gram-negative bacteria. This gram-positive bacteria, they have thick cell wall which is surrounding the plasma membrane. But what about gram-negative bacteria? This gram-positive bacteria, they have thick cell wall, and this gram-negative bacteria, they have thin cell wall. In addition to the thin cell wall, gram-negative bacteria also have an outer membrane. Okay, and the region between the outer membrane and the cell wall is the periplasmic space. We will discuss about it. Next is bacterial flagella. So these bacterial flagella, they are tail-like structures, and by showing the rotation, okay, bacterial flagella shows rotational movement and they can move through the surrounding media. Okay, so in response to chemicals, these flagella rotate and this movement is known as chemotaxis. Bacterial flagella are made up of protein named flagellin. Okay, this flagellin forms a long filament, and this filament is attached to the motor. Okay, the filament is attached to a motor by a hook. So you can see here this flagellin, which is forming a long filament, and this filament is attached to the motor, and this motor and filament is attached by a flagellal hook. So where are these eubacteria as well as archaea are found? What is their habitat? What is their habitat? So bacteria or the eubacteria, they are found in soil, water, or living in the large organisms which include E. coli and Bacillus species. Also cyanobacteria, which is under eubacteria. These cyanobacteria are blue-green algae. Now coming to archaea, the archaea are found in the extreme or the unusual environment like soil brines, hot acids, springs, bogs, etc. And archaea example, if we see, it includes sulfur bacteria, methanogens, and they are also some archaea which are found in the less hostile environments. Now coming to the next part, which is cell structure. So bacteria, they exist in several shapes: spherical, rod-like or helically coiled. Spherical are cocci, rod-like are bacilli, and helical is spirilla. Most of the time it is asked in MCQ which bacteria are helically coiled. So they are spirilla. Okay, so as we discussed, all bacteria are surrounded by plasma membrane, but they lack organelles. Okay, so plasma membrane are like the normal ones found in eukaryote. They consist of lipid bilayer containing proteins, which is similar with eukaryotes. Now, like eukaryotes, they lack the subcellular organelles. Okay, and these plasma membrane have infoldings called mesosome. Now, what is the role of mesosome? Okay, so these mesosomes, they are the site of DNA replication. Number one, they are the site of DNA replication. Next is what? They also, in the mesosome, they are also the site of some enzymatic reactions. Okay, for photosynthetic bacteria like cyanobacteria, for photosynthetic bacteria, they are the site of photosynthesis. Okay, mesosome contains proteins, they contains pigments which helps to trap the light and generate ATP. Okay, and the cytosol contains other molecules like enzymes, mRNA, tRNA, ribosomes, organic compounds, etc., which are needed for cellular metabolism. Now, in the cytosol, the chromosome is also present in what form? In a single circular molecule of DNA. And this single circular is condensed to form what? Nucleoid. So, remember, in prokaryotes, the DNA is present in the condensed form, where? In the cytosol. Because there is no membrane bound nucleus, so where will the DNA be present? It will be present in nucleus. Sorry, it is present in cytosol. Now this part is very important, the structure of bacterial cell wall. So as discussed, it is composed of peptidoglycan, which is a complex of oligosaccharide and protein. Now the oligosaccharide component. So what we discussed, it has an oligosaccharide component and a protein component. The oligosaccharide component, it has the alternating units of GalNAc, which is N-acetyl glucosamine and N-acetyl muramic acid. Okay, it has alternating units of GalNAc and NAM. Okay, and they are linked by what linkage? Beta 1,4 linkage. Very, very important to remember. GalNAc and NAM, they are linked by beta 1,4 linkage. Also see to the lactic group on NAM. Okay, just imagine on the NAM there is a lactic group. Okay, and on the lactic group, via an amide bond, there is an amino acid which is a D amino acid. So on the NAM, there is a lactic acid group. Okay, there is a lactic acid group here. To the lactic acid group, there is D amino acid formation. There here is the linkage of D amino acid via the amide bond. Okay, now this D amino acid tetrapeptide is linked to the, I write it clearly, to the D amino acid tetrapeptide. This is linked to the lactic acid via the amide bond. Okay. Now see another important point. that the adjacent parallel peptidoglycans, they are cross-linked through this tetrapeptide which is there, through this tetrapeptide side chains. How? By other short peptides. Okay, so what it is saying, that the adjacent peptidoglycans, they are cross-linked via this tetrapeptide by some short peptides. And this cross-linking, extensive cross-linking, that is D amino acid tetrapeptide of 1 NAM with the other peptidoglycan layer. Okay, this extensive cross-linking is giving the strength and rigidity to the cell wall so that they become very resistant to the proteases. Now this D amino acid tetrapeptide, they renders the cell wall resistant to proteases. Now as you can see the structure, this is a plasma membrane infolding which is known as mesosome. Okay, they have the flagellum which is made up of protein flagellin, and the DNA is condensed. DNA is circular and condensed, present in the cytosol. Okay, and the space between the outer membrane. So when outer membrane is present, that means which kind of bacteria is this? This is gram-negative bacteria, which have a outer membrane and a thin cell wall. Gram-negative bacteria have a thin cell wall, and the space between the outer membrane and the cell wall is periplasmic space. Clear? Now one important point to remember, that antibiotic penicillin, what it does, I told it targets the enzyme which helps in cell wall synthesis. But which step does it target? So it inhibits the enzyme that forms this covalent cross-link in the peptidoglycan. So what is this covalent cross-link? So this covalent cross-link was the linking between the D-amino acid tetrapeptide with the other peptidoglycan layer. So this cross-link is inhibited by penicillin. Because this cross-linking is the one which is making the cell wall more resistant. So this cross-linking is inhibited by penicillin, which is rendering the cell wall to become weak. Okay, the next important point, the beta-1,4 glycosidic linkage which forms between NAM and GalNAc is susceptible to hydrolysis by lysozyme. Okay, so this lysozyme, it is able to break the beta-1,4 linkage between N-acetylglucosamine and N-acetylmuramic acid. Okay, now so the bacteria we already discussed, they can be gram positive or gram negative, and this gram positive and gram negative name has been given depending on the stain they can take up or not. Okay, already discussed, this gram negative bacteria have a outer membrane which is external to the cell wall, and this outer membrane is very permeable to the large molecules, maybe greater than 1000 Dalton, because they have many porine proteins. Okay, now between the outer membrane and the cell wall is a layer known as periplasm, which is a space between the outer membrane and cell wall is the periplasm. And this periplasm, in this periplasm, there are proteins which are secreted from the cell. Okay, so whatever proteins the cell secretes, they are accumulated in the periplasm. Okay. Next, coming to bacterial flagella. So already discussed that the bacterial flagella are made up of a protein named flagellin. Okay, bacteria shows rotational movement of their flagella in response to the chemicals. Okay, so they can move through the extracellular medium towards attractants and away from the repellents to so-called chemotaxis. So movement in response to chemicals is chemotaxis. So these bacterial flagella, they are different from the eukaryotic flagella in two ways. What are the two ways? First is bacterial flagella are made up of flagellin protein. And in eukaryotes, it is made up of tubulin protein. The second difference is bacterial flagella rotates and the eukaryotic flagella bends. Okay, the first is it is composed of flagellin protein rather than tubulin, and the second is bacterial flagella rotates rather than bends. Okay, if we go through the structure of the flagella in electron microscopy, how does it look like? So it was seen that, if I write what are the parts, it was seen that there is a flagellar filament. There is a flagellar filament which is composed of 11 subunits, and these 11 subunits are arranged on two helical turns. Okay, and it seems like 11 blade propeller with a hollow central core. Okay, these 11 subunits are arranged in two helical turns and seems like there is a hollow central core. And the new flagellin subunits, they are added when the flagella grows. Okay, so flagella grows by the addition of new flagellin subunits to the end away from the cell. Okay, and where are these new subunits coming from? The new subunits are coming from the central core. Okay, now, so between the flagellar filament and the cell membrane is the flagellar hook. The second part is the flagellar hook. Where this flagellar hook is present, it is present between the cell membrane and the flagellar filament. Okay, between the cell membrane and flagellar filament is a flagellar hook which is composed of 42 or which is made up of 42 kilodalton. Okay, here it was 11 subunits. So are you getting me, guys? So this structure is simpler. I will show you the picture. So the second part is flagellar hook. This flagellar hook forms a short curved structure. Now coming to the third part is flagellar motor. Where is this flagellar motor present? This flagellar motor is also known as basal body. Okay, now this flexible hook, which is the second part, it is attached to a series of protein rings which are embedded in the inner and outer membrane. So look at the last point, which is the flagellar motor or the basal body. So they are the intricate assembly of the proteins. This flexible hook is I write here, they are the assembly of proteins. I will show you the picture how it looks like. Now see, the hook is attached to a series of protein rings, and these rings are embedded in the inner and the outer membrane. Okay, the hook which is there, it is attached with the several proteins, and these proteins are embedded in the inner and the outer membrane, and the rotation of the flagella, it is driven by protons through an outer ring of proteins. Okay, there is an outer ring of protein which helps in the proton movement, which is called stator. Okay, now this is similar to F1 F0 ATP synthase, which synthesize ATP. Now look at the structure here. So this is a cell wall structure of the gram-positive and gram-negative bacteria. So see here, gram-positive bacteria have a thick cell wall, and gram-negative bacteria have a thin cell wall, and in addition they have outer membrane which consists of several pores. Okay, now look at the structure. So see, it has a hook and the filament. Okay, the filament, after that filament is a hook which is connected to the motor. Okay, this is a region, this is a basal body, and this is a stator which helps in the proton movement. Okay, so guys, once again let's revise about the structure of the flagella. It has a flagellar filament, as you have seen in the structure. It consists of 11 subunits. Okay, these 11 subunits are arranged in two helical turns, and there is a central core. So new flagellin subunits, they comes out from the central core and they are added to the helical structure, to the helical turn, to grow the flagella. So after the filament is the flagellar hook, which is composed of 42 kDa, and this forms the curved structure. Okay, this hook forms a curved structure. As you can see here that this hook is forming the curved structure, and it is composed of 42 kilodalton, and this filament it has a central core and the 11 subunits which are arranged in two helical turns. After that is the basal body. So the third one is basal body or the flagellar motor. It is an intricate assembly of proteins, and the flexible hook is attached to a series of protein rings, and these rings are embedded in both the membranes. So look here. You can see here that this flexible hook is attached to the series of protein ring, L ring, P ring, as you can see here, and these rings are embedded in both the outer membrane and the inner membrane. You can see here in the inner membrane, here also the P ring is present, the outer membrane L ring is present, and this L ring is present here in the outer membrane side, and the P ring is present towards the inner membrane side. So they are embedded in both the membranes. The last point is the rotation of the flagella. Okay, in response to chemical, it is driven by protons through an outer ring of proteins. Okay, the stator is helping in the movement of the flagella, in the rotation movement of the flagella, and this stator, so this stator which is giving or providing the energy for the movement of the flagella. Okay, and this stator is made up of MOT A and MOT B proteins. So this is all about prokaryotic cell structure, the flagella of prokaryotes, the cell wall structure, and the cell membrane, the difference between the gram positive and gram negative bacteria. So that's it, friends. See you again. Thank you.